Good evening, everyone. We'll wait for 5 minutes, let others join, and then we'll start the session. Okay, so is my screen visible to all of you? Is the slide visible? Okay, I hope everyone can see my slides. Okay, so shall we start the session? Is the slide visible? Anyone can say yes.
Yes, ma'am.
Okay, good. Good evening, all of you. So, I myself is Tanishka, and I am teaching you the course Enzyme Science and Technology. The instructor of this course is Professor Vishal Trivedi. So, in this course, this week's session contains the information related to the delivery of the gene of interest into the host cell. How will you screen them, and how will you overexpress your desired gene of interest, or you can say the enzyme which you are planning to clone. Okay, so we'll cover all of these in today's problem solving session. So to start with, let's see our first problem today. Okay, so first is, what do you mean by competency of a cell? Okay, can anyone tell me the answer? What could be the answer? Okay, what is competency? Anyone? What do you understand by competency of a cell, of a bacterial cell? Why do we need to make them competent? Does anyone know? Okay, so first option is the insertion of a formed plasmid into the cell. Whether that is the thing which you understand by competency? Whether the first option is correct? Anyone? What do you think?
No, ma'am.
What about the second one? The chemical process of switching the gene?
Ma'am, I think fourth one is the correct option.
Yeah. Change in structure and permeability of cell membrane. Okay. So, competency meaning you are changing the permeability of the cell membrane. And why there is a need to do that?
To take up the foreign DNA from the environment.
Okay. Why can't a cell without being permeable or without any change normally can take up the DNA? Suppose one cell is there and you want it to take up the DNA. Whether the normal cell can take up the DNA?
No, ma'am.
Okay. Why?
Ma'am, because DNA is negatively charged and this charge on cell membrane is also negative. So
Hmm.
there is possibility that both will repel each
Hmm.
other.
Hmm.
That's why we need to make cell membrane permeable so that this DNA can be easily get inside.
Yes. So in the cell membrane, we have the phospholipid bilayer, right? This kind of bilayer, and this phospholipid is negatively charged. Okay? And the DNA of your interest, that is also negatively charged. Okay, this is also negative, this is also negative. Okay, and also DNA is hydrophilic in nature, and membrane is made of lipid. Okay, and then that is also one region normal cell membrane cannot take up this DNA. Okay, so in order to take up the DNA, the cell structure needs to be changed or make it need to be made permeable so that the foreign DNA can enter. And for that, you what you do, anybody can tell what is the approach that we use to make it permeable?
Calcium fluoride treatment.
Yes, CaCl2 treatment, and this calcium ions are positively charged. Okay, and these positively charged ion helps the DNA to bring closer in the proximity of the membrane, because what happens, suppose this is the membrane, this is the negative charge, and you provide the positive charge from the calcium ion. So you have the cell which is incubated with the calcium chloride, so that is surrounded by this positive charge, and now the negative DNA can come closer to this cell membrane. Okay, so it comes in the closer proximity. And then how does it go inside? There is one more step we need to do to take up the DNA by the cell. What is that step?
Gate shock.
Yeah, the pore needs to be opened, and that we do by giving heat shock to the protein. Okay, just a minute. Just give me 2 minutes. Okay, so heat shock needs to be provided. Heat shock needs to be provided so that the cell can, the spores can be opened and DNA can come inside the cell, and it can enter inside the cell, and then the DNA can be transformed, the cell can be transformed. So the correct option in this particular case would be the change in structure and permeability of the cell membrane. Let's see next. Okay. Okay. So next is what will be the order of the steps in the bacterial transformation. We just now discussed. So can anyone tell out of these four what would be the first step for us to do for the bacterial transformation? Out of these four, what would be the first step for us? Waiting for the answer. Out of these four, what could be the first step? We just now discussed.
Incubation of bacterial cells with calcium chloride.
Yes. First will be incubation so that it can become permeable. And then,
Heat shock.
yes, second we just talked, we have to give heat shock to the cell so that pores can open for a small period of the time, only transient opening of the pores, and then DNA can come inside. And then we have to provide a normal condition or a cold condition so that the pores can again close and cell wall can become normal, cell membrane. And then what we do next?
Recovery of the cells.
Yes, then third step will be recovery. That means we add some amount of media so that, and we allow some times for the cells to grow and recover. And then what we do? Fourth is antibiotic selection, right? Once the DNA has been taken up, now we will select our cells which are considering transformed in an antibiotic selection plate, because we generally transform our plasmid, and our plasmid should have a gene that codes a gene that will provide resistance against one particular antibiotic. Suppose we have ampicillin antibiotic resistance gene, and this plasmid, if we are putting inside a bacterial cell, then this particular bacterial cell can grow on a plate LB plate which have the ampicillin, and the cell which has not taken up this plasmid will not be able to grow because it doesn't have any cope-up mechanism against ampicillin. People are joining. Okay, so this will be the order of the steps. First, we'll incubate the bacterial cell with calcium chloride so that it can become permeable, and then we'll add the DNA to that particular cell, and then allow the DNA to come to the surface of the bacterial cell, and then we'll give a heat shock so that a transient opening of pore can happen and DNA can come inside the bacterial cell. Then we'll give some LB media to provide to the cell so that it can grow and recover, and then that particular bacterial cell we spread on an antibiotic selection plate so that we'll get the transformed bacteria only. Okay, and then another thing will be transformation of yeast cells. So there we use one transformation mixture. Can anybody tell what is that transformation mixture? Anyone? Okay, so can anybody tell the just now I mentioned one method for bacterial transformation and making it competent with calcium chloride. Do we do the same thing with the yeast cell transformation as well? Yes or no? Do we do the same method for yeast cell transformation as well? Anybody know? So the answer is no. We do not use the usual calcium chloride method to making that yeast cell permeable. Instead of that, the transformation mixture which I am talking about in yeast cell transformation would contain lithium acetate, and another is PEG solution which is polyethylene glycol, and ssDNA. These three things are needed for the bacterial cells transformation to make yeast cell transformation to make yeast cell competent. Okay, so the function of these individually, LiUC lithium acetate just function which is doing uh, just the function it does the calcium chloride, which is in case of bacterial transformation lithium acetates provide permeability. It makes the yeast cell permeable. PEG works as a adhesive reagent you can say. It works as an adhesive reagent. It brings the DNA and the yeast cell in close proximity. And ssDNA, it protects your particular DNA of interest from the endonuclease activity of yeast cell, and also it inhibit the uh, non-specific sorry it blocks the non-specific surfaces on yeast cell. Okay, so that your particular DNA of interest can easily go into the yeast cell and can be transformed. Okay, so this is the mixture we use for the yeast cell transformation, and the individual function of them is listed here. Okay, and similarly for the mammalian cell, and in other cases also sometimes we used electroporation also. Electroporation, there we give a small electric treatment, but at a particular voltage or something, and so that for a transient a time, for a little time, for few 1 or 2 seconds, this wall or membrane can open and DNA come inside, and then we allow the recovery of the cell and grow them, and then we provide the selection medium and everything so that it can be selected, the transformed cell can be selected. Okay, next if we'll see. Okay, so in the class you have also, in the note it is provided how to calculate the transformation efficiency and everything. So this is a question related to that particular topic. So if you have a plasmid sample that has the concentration of 100 nanogram per microliter, and you have added 2 microliter from this plasmid into 100 microliter of the competent E. coli cells, okay, and after those steps of bacterial transformation you have added 900 microliter of media. After 1 hour incubation, you have plated only 100 microliter from there on an antibiotic containing agar plate. And then next day you have got 95 colonies on your plate. So my first question would be, how much DNA was added to these competent cells? Tell me the answer. The concentration is 100 nanogram per microliter. So how much exactly, what amount you have added to the competent cell? Very easy question it is. Everything is just written. Just read the question and tell me the answer. The concentration is given 100 nanogram per microliter, and you are adding 2 microliter from this plasmid. So how much actually you are adding to the competent cell? That is the question. How much DNA?
200 nanograms.
Yes, you are adding 200 nanogram DNA. Okay. And then the second question is, how much DNA was spread on the plate? Okay. So to calculate this, you have a formula. DNA spread on plate, you can calculate by volume which you have spread it into DNA amount that you have transformed, and then total volume of transformation. Okay. So what will be the volume spread? What would be the volume spread in this case?
100 microliters.
Yes. 100 microliter. And what was the DNA amount taken?
200 nanograms.
200 nanogram. And the total volume of transformation?
900 microliters.
No. You have added 900, but you already had 100 microliter of the competent cell. So 100 plus 900. Total
One,
volume is,
1 ml.
yeah, 1 ml, 1000 microliter. Okay. So DNA spread on the plate will be 20 nanogram. Okay. Or you can say, if you want to say in microgram, you can say 0.02 microgram, because transformation efficiency we calculated microgram generally. Okay. So this is the DNA, amount of DNA that is being spread on the plate. The next question is, what will be the transformation efficiency? Does anyone know the formula? How you calculate the transformation efficiency?
Ninety-five divided by 0.02.
Yes, so the formula is, to calculate the transformation efficiency, you calculate by number of CFU divided by DNA spread on the plate. Okay. And transformation efficiency, the unit of transformation efficiency is number of colony forming units per microgram of DNA. So for to calculate that, we just divide the number of colony that we have observed and the DNA we spread. So 95 divided by 0.02. So your transformation efficiency will come, I think, can say 4.75 per microgram. Okay. So that is a general calculation. If a question comes, what would be the transformation efficiency, how much DNA you spread and everything, I think you can do. Okay. Suppose you have got the transformation efficiency and other thing. So it can be asked how much DNA you have started with. Okay. The question can come like that also. So the basics you need to understand and the formulas you need to remember so that you can do the back calculation as well and get the answer. Okay. So here are the 4 plates where a transformation has been performed in E. coli bacterial cells, and the plasmid harbors a ampicillin resistance gene, and these are the results obtained after the transformation. Okay. So first plate here is normal LB plate without antibiotic, and where this ampicillin resistance plasmid is transferred and plated. Okay. Another plate here showing the LB agar with ampicillin added, and the ampicillin resistance plasmid also being added here for the transformation. Another here is normal LB plate and the bacterial cells without being added any plasmid. No plasmid is added here, and just normal transformation is being performed and plated on this plate. And here LB agar with ampicillin and no plasmid is added, and then the cells were being spread on this plate. These are the 4 kinds of plate and different scenarios. So the question here is, on which Petri dish only transformant cells will grow? Out of these 4, which one is the particular Petri plate which is showing the transformants? Okay, if we'll see the first plate, okay, this is LB plate only without antibiotic, and here you have transformed the plasmid, okay, and you have got a lawn kind of structure. So do you think here you have got all the cells which are transformant only? Whatever is growing on this plate, whether all of the cells are transformant? Yes or no?
No, ma'am.
Why?
Ma'am, because in this plate, antibiotic is not present, so non-transformants will also grow.
Yes, exactly. Without antibiotic, non-transparent cells will also grow. Okay. And what about the second plate?
Ma'am, in this plate only transformant cells can grow.
Yeah. Because antibiotic selection is being provided and ampicillin resistance plasmid is also there. So the only that cell which has taken up the plasmid will grow and other cell will not grow. So this is the answer of your first question, that this is the particular plate where only transformant cell will grow. And the third plate where LB agar is there, no plasmid is added, still the cells are growing. That means these cells are just normal E. coli bacterial cell without any plasmid, and they can grow on normal LB agar plate because they don't have any antibiotic resistance, and this plate is also normal LB plate and they can easily grow here. And the second plate where no plasmid is added and just the cells, and in that scenario also cell will not grow because it has no plasmid so that can provide the resistance against ampicillin. So what would be the answer for this particular second question? Which of the plate is used as a control to show that non-transformant E. coli will not grow in the presence of ampicillin?
Plate 4.
Yeah. Yes. Plate 4. This work as a control that our cells are clean and they have no background of no background that will grow on the ampicillin plate. So whatever you are getting on this plate 2, these are the true transformant cells. Okay. So So all these transformation and everything you are doing so that you can overexpress a particular gene and get the protein or enzyme of your interest. So in that scenario, an expression vector will be needed. And what are the it contains a sequence which is not normally found in other vectors. So what is that sequence out of these four?
Ribosomal binding site.
Yes, because other three can be present in a normal vector also, but if you want an expression vector where you want transcription, translation to happen, you need ribosome binding site. So the correct option will be ribosome binding site. Okay. Yeah, so talking about the expression vector. So,
Ma'am,
yeah.
there's a doubt. Like in cloning vectors, do we have this uh, promoters and
Hmm.
like in cloning vectors, do we have promoters?
Yes, promoter will be present. Like suppose a normal expression vector I am talking. Suppose this is a PET28A vector. It will have a promoter. It will have a gene of interest which you have cloned here so that you can overexpress it, and other resistance for any particular antibiotic, suppose CAN resistance, and or origin of replication. All these sites will be present in a normal vector. Okay, so to overexpress one particular gene, what we do, we use a particular kind of expression cells. Okay, expression cells. Okay,
Yes,
for
ma'am.
example, BL21 D3,
BL21,
yeah,
D3.
Rosetta, and everything. Okay, so the plasmid we are not changing. For the initial purpose, we keep the same plasmid here. For the cloning, for screening and everything, we use our normal cloning cells like DH5 alpha or top 10 cells. Okay, so when your plasmid will be present in these cells, it will not allow the translation or transcription of the protein. Here your protein will remain intact, sorry, your gene will remain intact. Transcription will not happen in these two cells. These are the cloning cells. Okay, in order to overexpress the protein, you need, or gene, you need to put this plasmid in this expression cell. Okay, and what exactly happens in the expression cell, because just now you asked whether this have the promoter. Yes, the vector will have the promoter, and generally we put a T7 polymerase promoter, for which the transcription factors will not be present in this two cells. The transcription factor to activate this promoter will be provided by this expression cell. So suppose in BL21D3 example I am taking, now your plasmid is confirmed and everything, you'll put this particular plasmid here in this BL21D3 cell. Okay, with the promoter and your gene. Okay, so here in the chromosome of this particular expression cell, you will be having T7 RNA polymerase. Okay, These are the engineered expression cell which will harbor T7 RNA polymerase in their chromosome, and that too in silenced condition. They have a repressor gene as well. This repressor gene, which under normal condition does not allow this T7 RNA polymerase to become active. It will always repress it. Okay. So when you provide IPTG, which is nothing but a work as an inducer. Okay.
Inducer.
Yes. So this IPTG, when provided, it can block this repressor, and thus this RNA polymerase will be active and will go to this vector and allow the transcription of your gene to happen, and then further translation and other machinery can carry on. And in that scenario, your protein will be expressed. Okay.
Ma'am, that means the vector that we have transformed into the cell previously, it is always containing this ribosomal binding site in the
Yes,
it
yes, yes,
is
yes.
in the expressing cells.
Yeah, it is the expression cell that provide the transcription factors.
Okay.
Hmm. Yeah, vector we do not change. We keep the particular expression vector. We just change the cell for our purpose. If we want the protein to overexpress, then we use this expression cell. If normal cloning and propagation of plasmid, we use this TH5 alpha and top 10, where the gene will not be transcribed.
Okay.
Okay. And yeah, and also when we do perform these experiments of overexpression and everything, what we do, we and suppose we have the primary culture with us. Okay. And then from this primary culture we again inoculate a secondary culture. Okay. And then we allow the this secondary culture to grow to up to an optimum OD point. Sorry. Okay. Yeah. So we allow the secondary culture to grow to zero point 0.6 to zero point 0.8 OD. Okay. And when the our OD reaches to 0.6 to 0.8, then only we add the IPTG to the cell. Okay. And then we allow the induction to carry on, like 4 hours, sometimes 16 hours, sometimes 24 hours, different time of induction, so that our protein can overexpress. So can anybody tell why we do not add IPTG at the very beginning itself? Why we allow them to grow at this particular OD?
When since we have changed the media, therefore
Hmm?
the cells will first acclimatize in that media,
Hmm?
and when it, initially it may be in the lab phase. So
Hmm?
when it will be suitable for the medium, when
Please carry on.
when it reaches to the OD of zero point 0.6 to zero point 0.8, that means the cells are actively dividing. That means they are in mid-log phase. So
Hmm.
at that time only the cells will be actively dividing, and their entire mechanism, their enzymes and everything will be working properly. So at that time we put IPDG so that our cells can be overexpressing the protein of interest.
Yeah, so
Which we
that
have
we
seen.
can add the initial also, like as soon as they grow and they start IPTG start acting, and the protein will start overexpressing. We can do at the initial phase also. Okay. But the reason which we, when we not use the IPTG in the beginning, is because you are expressing a heterologous protein inside a bacteria. You are giving a eukaryotic protein, because in general what we do, some foreign DNA we add, right, the foreign gene which doesn't belong to the bacteria. Okay. So if from the beginning bacteria will start producing this gene which for bacteria has no use, right? Bacteria has no use of this particular protein which is being expressed inside it. So bacteria will have a sense that this particular protein is of no use, and thus it is a toxic for bacteria. Okay, and when that scenario comes, the bacteria will not grow further, it will start dying. Okay.
Yes, ma'am.
Yeah, so that is the reason we allow the bacteria to grow to particular log phase. You get enough amount of bacterial cells in that particular OD 0.6, 0.8, and then you add IPTG and allow the cells to grow for further 4 hours, 5 hours, or so on time. Okay, so now whatever cells you have grown will for sure produce the protein. Okay, and then after that, even though they are not like other bacteria will not grow, like suppose this will not further divide and give the other population, the next population, even though it will not grow, but the population which is already there, which has it has already expressed the protein. Okay, and that protein you will take up by harvesting the cells, and then you proceed with your experiments, purify the protein and everything. Okay, that is the reason we add IPTG after growing it to a particular log phase. And one more reason why we use like this IPTG. This IPTG is an analog of lactose, right? Why do not we use lactose directly?
Is it because ActoGene is easily permeable to the cells?
No, no, no. Okay, so because we are using the lac operon system and everything, we need lactose, but we do not use lactose. Instead of that, we use an analog of lactose that is called IPTG, because this IPTG is you can say it cannot be consumed by bacteria. Okay, non degradable, because lactose if you will provide, bacteria will use it up. Okay, and break it into the further smaller biomolecules. Lactose will be utilized by bacteria, but IPTG cannot be utilized by bacteria. Okay, so if you provide lactose, bacteria will immediately break it and make the smaller components. Then again you have to provide more lactose so that more induction can happen. Okay, so to avoid those particular things, we just directly give IPTG, which is an analog of lactose, which is non-consumable for bacteria, and you can keep the induction culture for any long time which you want, and it will not be insufficient for the cell. Okay, understood? Okay, you got the point here? Yes, no. Shall I go ahead? Anyone, if you have not understand what I just said, you can ask me again. Okay? Or shall I move ahead? Okay, let's go to the next point. Hmm. Okay. Yeah, one more thing I wanted to say here. Okay. So you are expressing a foreign gene, a eukaryotic protein into a prokaryotic cell. Okay. And suppose it has a gene sequence. Okay. It has particular codon, and sometimes it happens like one gene can be coded by many different codons. You know that, right? Everybody knows that. The degeneracy
Yes,
of
ma'am.
codon, right? So suppose whatever codon is present inside your eukaryotic gene which you are trying to overexpress in bacteria, and that for that particular codon the anticodon tRNA is not there in the prokaryote, because suppose for leucine, the leucine amino acid, your eukaryote uses a different codon. But inside a bacteria, bacteria uses another codon for production of leucine, and bacteria will harbor that another tRNA only for that leucine. But that tRNA cannot identify the bacterial, the eukaryotic codon. So in order to do that, so in that scenario, the protein will not overexpress because of the lacking of that particular tRNA, okay, inside the bacteria. So in order to solve that problem, the other strains of the bacteria are designed. For example, Rosetta, another P. lyes, and other different many cells are there where extra tRNAs are added. Okay, that will code for another eukaryotic codons, and thus your protein overexpression can take place smoothly without being hampered. Okay, so
Yes, ma'am.
yeah, so that is the point, codon biasness or degeneracy of codon, that is also important in the selection of expression cell. Okay, next is the blue white selection. Okay, so what would be the correct option here for blue white selection? Okay, for what it is used? To test the presence of plasmid in bacteria, to reveal the identity of a cloned DNA fragment, to express the product of a cloned gene, or to test the presence of a cloned insert in a plasmid. What would be the option?
Ma'am, D.
B.
D.
D. Yeah, to test the presence of a cloned insert into plasmid. So we'll see here for blue white screening. Okay, one cell which is non-transformed, so it will to definitely not grow an antibiotic resistant cell. Another, there are two possibilities: transformed with non-recombinant, transformed with recombinant vector. Okay, like here the LAC-Z, inside the LAC-Z is the site for cloning the insert. Okay, so if your gene is cloned into this particular insert, so the LAC-Z won't be active because it is now disrupted. Okay, so inactive. Okay, so if the LAC-Z is active, it can convert the X-gal substrate to a blue color product with the help of beta galactosidase. Okay, and if the DNA insert is present, then the beta gal will not be active, and X gal cannot convert into a blue color product. So DNA insertion is indicated by white colonies, and the blue colonies will be the non-transformed or non-recombinant colonies. Okay, so for that particular purpose, we select our colonies on an agar plus antibiotic plus IPTG and will provide the X-gal substrate, and then we get this kind of result. So the white colonies are the true transformed colonies where insert is being cloned, and the the blue colonies are the where the lag-Z is intact and the DNA insert is not cloned. Okay. Okay, so we'll go to the next question. Yeah, which of the following is not a potential problem associated with expressing a eukaryotic protein coding nuclear gene in prokaryotic cell, which is not a potential problem. Okay, out of these four. So first is lack of intron splicing mechanism in prokaryote. Whether this could be a problem in expressing a eukaryotic gene in prokaryotic cell?
No, ma'am.
No, why? It is a problem. Suppose if you are giving a eukaryotic gene which has intron in it, it cannot express itself inside a prokaryote because prokaryote lack the intron splicing machinery. Got the point?
Yeah, yes, ma'am.
Yeah.
Got it.
Yeah. Prokaryote doesn't have the intron splicing machinery. Okay. So if you have any gene of interest that has intron in between it, inside the prokaryotic cell, it will not overexpress. So you have to take your gene that without intron and then clone it into the desired vector and then try to overexpress in the prokaryotic cell. Then the expression will take place. Okay?
Yes, ma'am. I think that's why we take mRNA and then we make cDNA and then clone it.
Yes, exactly. If you have some gene in your mind that you want to clone and overexpress in bacteria, but it has intron, so in that case you have to isolate the RNA, make it to the cDNA, and from that cDNA you have to prepare that particular gene of interest to clone. Okay?
Yes, ma'am.
Yeah. Okay, so this is a wrong option. Then stability of mRNA in prokaryotic cell. Whether this is associated with the expression problem in prokaryotic cell. It can be a problem, right? There are chances that while transcription, the mRNA which is being produced, it is not stable in the prokaryotic cell. Okay, because sometimes we do that, we are giving the IPTG induction, the gene is there, still the protein is not being formed because the RNA itself is not stable into the prokaryotic system. So to make it stable, we have to do some changes. Like you have must have heard the induction at lower temperature, right? Or less IPTG concentrations and everything. These changes we do so that RNA can be stable. Okay. So lower temperature provides stability. Okay. Lower temperature provides stability, and thus how, like suppose if the normal temperature of bacteria like 37 degree, the protein you cannot see, you can try, you can, there is a chance that the mRNA which is being formed is not stable. You try to decrease the temperature and then provide the induction and then see whether you are able to see some amount of protein or not, because that makes the RNA and protein stable, the lower temperature. Okay, so this is a problem which you should work on if the protein is not overexpressing in a prokaryotic system. Okay. Third is differences in the transcriptional signals between eukaryotic and prokaryotic cell. Okay, this is also a possibility. Okay, the transcription factor signals between eukaryotic and prokaryotic differs. Okay, so sometimes these are the cases, like just now I have talked those codon biasness. Okay, so you have to provide those extra tRNA so that the overexpression can take place, right? The transcription and translation can happen. So the transcriptional signals and factors are also required. Now the fourth one, the differences in the translation initiation codon used by eukaryotic cell and prokaryotic cell. Whether this is a potential problem while expressing a eukaryotic gene?
No, ma'am.
No, right, because the initial codon too, it will be same, the methionine. It is not talking
Yes,
about the
ma'am.
other codons. It is the initial codons. This is not a problem. So for that question, this particular option is correct. Okay. Let's go ahead. So for screening, you also heard about the sorry, I have written repressor. It is reporter. Reporter. Yeah. A reporter gene. So what does it do? Out of these four options, what would be the correct option?
See.
Yes, allow the gene expression to be readily measured. If you have a reporter gene, you can easily measure your gene expression. Okay. Uh, suppose, okay. Yeah, let's see here. The next question is, which of the following is incorrect about a reporter gene? First is gene whose phenotypes can be assayed in a transformed organism. Whether this is correct in regards of reporter gene?
Yes, ma'am.
Yes. Reporter gene is used so that it can give a particular phenotype that can be assessed in a transformed organism. Next is beta galactosidase gene is an example of reporter gene. Correct or not?
Yes, ma'am.
Yes. Just now in the blue white screening I told you LAC-Z. So it produces this beta galactosidase which acts on the X gal and converts it into a blue product. Okay, so thus it works as a reporter gene, that if that gene is being expressed, the X gal will convert it to blue color, and that's how you can do the screening. So this is also a correct option. It is a reporter gene. Another is test gene, that is fused to the upstream region of the cloned gene.
It should be in the downstream region of the clone gene.
Both are correct. Actually, upstream is also you can do, like if you have a gene of interest here which starts from the ATG, in the same frame you can clone your reporter gene as well, maintaining the frame like ATG and then the next three codons and then the three, like this. Like addition of this particular should not disrupt the uh, frame, ORF frame to this one. So upstream you can also put. Okay, like luciferase assay, there I think it is in the upstream, and with the luciferase you can just see the emission and everything after the lysis, and that will also give the indication whether your gene of interest is expressing or not. Okay, I hope it is clear. Yeah, and other than that, for the screening, you have heard about that particular some complementation and something, I think you have read, like suppose
Alpha
you
complementation.
yes, so where you, like suppose a plasmid is harboring his his 3 gene. Okay, and that particular his gene is mutated into the cell, in the yeast cell, in their chromosome, the his gene is not active. So to grow this kind of cells, like suppose the empty cell without plasmid, you have to provide histidine in the media, right, in the plate or something. Okay, you have to provide histidine. But if you have the plasmid in it, without this histidine containing medium, your cells will grow. So that's how you select these kind of clones or something, transformants. Okay, okay, so next is most common reporter gene whose product can be directly visualized in transformed cell. Okay, what could be?
GFP, ma'am.
Yes, GFP. If you visualize the cells under microscope, you'll be able to see a green fluorescence, which is because of GFP. And in order to do that, you have to make a fusion protein, right? Fusion protein. Okay, like in a plasmid, you have cloned your gene, and same with this, you can clone your GFP gene. Okay, and then you make the promoter active. So with your gene, like this total fusion will express together. Okay, so by seeing the fluorescence, you can say that yes, your gene of interest is also being expressed with GFP. Okay, that's how you see whether your gene is being expressed or not, with the help of microscope, directly visualizing the cells. Yeah, also you can try the sequencing method, right? Suppose you have like one plasmid, you put one like ligation of a vector and an insert, right? That ligation mixture you have transformed in a bacterial cell, you have got many colonies. Now you started screening whether the vector and insert got ligated or not. So you uh, isolate the plasmid with many colonies and send the plasmid for sequencing and allow the sequencing to carry on. And then by sequencing also you can analyze whether your gene of interest is present in that particular colony or not, right? So here dideoxy DNA sequencing is depend on which of the step out of these four. Can you tell me?
Chain termination.
Yes, termination. Because dideoxy, like our DNA is deoxynucleotide, right? And that means at the 2' OH of the DNA, that hydroxy is removed. But in dideoxy, the 3' OH is also removed. Okay, so it cannot carry on the next addition of next nucleotide, right? So in the sequencing you give some sort of primers and then allow the elongation of that change, but and you give dideoxy, suppose it be these particular with different tags. Suppose one will be green, one will be blue, red, yellow, different color it will be tagged, and you give normal DNTP as well. Okay, allow the sequencing to happen. Suppose your primer is bind, and okay, and here you give the all the mixture, and suppose you have, and at G, if your this particular uh, is being incorporated here, then the next one, that elongation will not take place. Your chain will be terminated at this particular point. Okay, and the same kind of reaction will happen. You'll get different fragment. Like suppose some will amplify till here, another will amplify for some longer stretch and everything, and this you allow on a very concise electrophoresis method, and that will detect the order, like first, second, third, fourth, like that. It will get the order of the nucleotides, and that's how you get your sequencing result. Okay, and you know after which nucleotide which one is coming, which one is coming, like that you get a particular map of the nucleotides one by one, and that you can compare with your original expected gene of interest, like what you are getting after the sequencing and what is the sequence you are expected to be there, and that you can align and say yes, your gene of interest is present into that particular transformed colony. Yeah, so what do you understand by adding a tag when cloning a gene? You have heard of many tags, right? What kind of tags you have heard?
Ma'am, for purification purpose, we a tag.
Mm-hmm. Like which tag?
Ma'am, histidine tag, GFP tag.
Hmm. Yes. Yes. You use 6 histidine residue, that is also a tag. You use GST tag, GFP tagged, HA tagged. These are the tagging you use, and each for different purpose, right? Just now you said that histidine tag you use for the purification of the protein, right? GST tag also for the purification, and GFP and HA tag also you use so that you can identify your protein. Like for GFP you can just see in the microscope and you can see that yeah, your protein is also there because the GFP is present. HA also you can identify by using anti-HA antibody also you can identify your protein of interest. Okay, so these are the tagging you do while cloning a gene. Okay.
Ma'am, is HA here hemagglutinin protein?
Yes, yes. Hemagglutinin. Okay. Okay. So yeah. So how can you incorporate a tag during cloning? Anyone know?
By introducing codons of that particular tag.
Hmm. Yeah, and you need to maintain that ORF as well.
Yes, ma'am.
Yes. And suppose if you want to do the tagging in the C terminal of the protein, then what you do? Like this is your protein of interest, N terminal, C terminal. So if you want to do the tagging in C terminal, will you just put your tag just after this ending of gene? Is there anything else you need to remember while doing this?
Ma'am, that stop codon should be at the end of his tag.
Yes. So the stop codon of your gene you need to remove. Okay? This place should be devoid of stop codon so that your tag will come just after your um last amino acid of your gene is being ended. Then immediately your tag transcription should be started. Stop codon should be removed and stop codon should come at the end of this tag, right? So that is the only thing we keep in mind. So whenever you do particular cloning, tagging and everything, if you have to use a C terminal tagging, you need to remove the stop codon from your gene so that in the C terminal in a frame your tagging can come. Right? And if you want to do just N terminal tagging, no problem. You can just no need to remove anything or add anything. You can just add your tag in the front and do the cloning normally. No need to remove the stop codon also from here. Okay. Stack. Okay. It's clear to everyone.
Yes, ma'am.
Yeah. And yeah, we just now talked about the histidine tag. So it helps in the protein purification. And how exactly it helps? How does that purification step is being done? Any idea?
Ma'am, we use nickel NPTEL column for purification of stack proteins.
Yeah. What does nickel NTA do?
Ma'am, nickel NPTEL is basically nitrile triaminic acetate column, which basically binds at histidines.
Hmm. Yes. Histidines. So if you lyse the bacterial cell, and if you lyse it, you'll get all the proteins of the bacterial cells, right? Including your desired protein. All the proteins will become like this is your desired protein, and other proteins will also come. So in order to take only your protein of interest, you need to apply the purification step. And for the purification, you need to use, for histidine tag protein purification, you need to use nickel anti beads. Okay. And this nickel anti, it binds to the 6 histidine residues which are present in your gene of interest or protein of interest. Okay. And yeah, and then after it binds to your protein, you run it through a column, suppose. Okay. Here you have your nickel anti beads and all the cell lysate. So your protein will be bound to this particular beads, and Thus, when you collect the flow through, okay, like you collect in another tube, your remaining flow through you'll collect from here, okay, and you are left with only beads where your protein is bound to those beads. Now, to remove the protein from the beads, to get the protein out of the beads, what you do?
When we use imidazole buffer
Yes, and what does imidazole do?
for competitive emission. When imidazole bind to this nickel anticolumn more strongly than histidine tag, and based on the competitive
Yes,
exclusion, it removes the protein that bind to the
yes,
column.
yeah. Nickel anti imidazole binds to nickel anti more efficiently than your protein of interest. So you use the imidazole in high concentration so that your nickel anti leaves your protein and bind to the imidazole. Now your protein is free and you can elute your protein of interest, and all the other bacterial proteins you have already removed at this step, and now your particular protein of interest will be eluted and you'll get pure protein from the bacterial cells. Okay. Next. So suppose you have overexpressed an exogenous protein in a cell, and if you want to determine if that protein is in fact overexpressed or not, what would be the best technique for you to use out of these listed here?
When we can simply use electrophoresis
Sorry, your
and
voice is breaking.
polyacrylamide gel electrophoresis.
Yeah, yeah. Please come again. What do you say? Please say again. I couldn't hear you the first time. Please.
Then we can simply use polyacrylamide gel electrophoresis.
Yeah, polyacrylamide gel electrophoresis. There you can see, like just now I said, no, polyacrylamide gel electrophoresis will, there will be, you lyse the cell and run all the content of the cells on a polyacrylamide gel. Okay, SDS page. There on the gel you'll get many proteins, many. So just to see like whether your protein is there or not, you have to do Western blot, right? You have to use specific antibody against your protein. You understood my point?
Yes, ma'am, in order to confirm that the
Yeah, in
overexpression
order to
protein is
confirm.
ours.
Yes. Exactly, because suppose if at that position whatever band you are saying normal on SDS gel, that could be of your host protein also of the same size. To be sure whether that is your protein of interest, you have to use Western blot so that you give an antibody against your protein that will bind to your protein, and then using the secondary antibody and the chemiluminescent you will develop the blot, and if you get a blot at the desired position, that you can confirm yes, your protein of interest is being expressed in this cell. Okay. Okay, so talking about the Western blot, so what do you mean by transfer in Western blot protocol? There is one step called transfer in Western blot protocol, right? What do you mean by that?
Option B.
Sorry?
Option B.
Yeah, move proteins from the SDS page to nitrosolidose membrane. Yes. So for Western blot, you just first simply run your protein here on the gel. You'll get many different all the cell proteins. Okay, and then from this gel you transfer the proteins to a membrane which is called a nitrocellulose membrane. Okay, from the gel it is being transferred to the nitrocellulose membrane just by giving those positive negative charges. All these proteins are negatively charged. You move them from uh, uh, towards a positive pole so that the all the protein content from gel is transferred to this membrane. Okay, everything is now transferred to the membrane. But this membrane protein will only be visible when you see with the Ponshu stain. Without any particular stain or everything, the membrane will look empty. Okay, no proteins will be visible. You have to give antibodies and the chemiluminescent so that you can see the desired band. Okay, so yeah. So why we are adding the secondary antibody while performing Western blot? Any idea?
To ensure that the primary antibody binds properly to the sample.
Mm, yeah. Okay. Yeah. Block any interfering noise coming from the membrane. No, this is not the function of the secondary antibody. Allow for detection of protein sample. Separate the sample from other protein. No. Ensure that primary antibody binds properly to the sample. No. No, not this option. Not this option. The option correct here is allow for the detection of the protein sample. Because your secondary antibody, okay, so suppose on the membrane you have your protein of interest here, okay, like many other proteins also, but your protein of interest, so your primary antibody binds here, right? And then for then you give the secondary antibody. The secondary antibody binds to the primary antibody. Okay, and this secondary antibody is conjugated with the horseradish peroxidase. Okay, and this only help for the detection of the protein sample. Okay, it does not ensure that primary antibody binding or not, right? Primary antibody has already bound to the protein. You are giving secondary antibodies so that you can detect, because primary antibody doesn't have any signal to give the band. Okay, secondary antibody has this particular signal so that the protein sample can be detected. Okay, so secondary antibody is conjugated with this HRP, horseradish peroxidase, and then you provide a substrate called luminol, and then with the help of hydrogen peroxide, luminol is getting oxidized by this peroxidase. Okay, oxidized, and that oxidized intermediate will um, show the band of the protein on the Western blot when provided with the chemiluminescent. Okay, so after these particular steps you will be able to see one particular band in the Western blot. That will be the specific band where your primary antibody is binding. Okay, okay, am I clear? Any doubt, anyone? Okay, so these are the questions, sample questions for that week's content. If you have any other question, you can ask me now. We also use blocking step in the Western, right? Why do you do that? The blocking step?
So antibody does not bind to the rest of the membrane or any other protein.
Yeah, to avoid the non-specific binding of the antibody, right? Yeah.
Yes, ma'am.
Yes. Okay. Other than that, any doubt you have? Any doubt in any of the questions you can ask me. We have time. See. Okay. Any doubt? We still have the time. We can
Ma'am,
discuss. Yes. Hmm?
ma'am, for example, we are using this Swiss-tag method in order to purify the protein. But we
Hmm.
don't know at the time of folding, there are chances that C terminus or N terminus of the protein, there are chances that it is hidden inside of the protein. So, in that case, like, how will we be purifying our protein if we are using this tag?
Yeah, yeah. Listen, that 6 histidine residue is a small stretch which can be commonly present in any of the protein, like in any of the host protein,
Yes,
and
ma'am.
yes, and any of the, like, suppose you are saying inside a gene of interest. Inside of gene of interest, no harm, but it there are chances that those
No, no,
hysts,
no,
yeah, that can be
I'm saying
present
that
in
there
other
are
bacteria.
chances that at the time of folding of the protein,
Hmm.
there are chances that the C terminus or N terminus, wherever our this tag is present, there are chances that it can be hidden inside the structure of the
Yes,
protein itself.
exactly.
And
There
at
are
that
chances.
time, if it is not exposed, it will not bind to the
Hmm.
column, then how can we, I will purify the protein?
Yeah, yeah, I know. So that's why, like, suppose there are chances because of this his tag, your folding is being affected. So suppose if you have put your his tag in the N terminal, you can try putting that to C terminal. If both of the N terminal, C terminal, in both of the cases the his tag is not being exposed, then in that case you have to change the tag. You have to use another vector with another tag, because anytime you do, you will not be able to uh, purify the protein, right? Or if you can do, you can provide denaturing condition, okay, so that your protein becomes
Okay.
linear, and that, so that then you can, then his tag will be obviously exposed because of the denaturation of protein, and using those denatured condition you can purify and try to renature quickly, okay. Or if your
Okay,
work
ma'am.
doesn't require a, like, functional protein, you just need your protein to purify for particular stuff like antibody generation purpose or anything, so in that case denatured protein will also work fine, okay. So no need to get a pure form of protein. But if you want to perform any biocatalytic assay, right, you want to check the activity and everything, in that case you need to have the pure protein or the functional protein, the folded functional protein, and in that scenario you have to change the tag so that you can purify the functional protein, active protein, right, without denaturing it.
Okay.
Yeah. Okay. Anything else? No, ma'am. Okay. You understood, Sakshi, the questions and everything? Okay. you can ask me if any doubt is there. Like if you're not able to understand at any point, you all can stop me and I'll explain again. Okay? Okay. Samiksha, Christina, you both clear with the session?
Yes, ma'am.
Okay. Good. Okay. Okay, so no further questions, so we can end the session. Okay.
Thank you, ma'am.
Okay. Thank you. Good interacting with all of you. Thank you. So we are closing the session now. Thank you.